Hello there, students from BYS School. This is teacher Krishna, and welcome to our biology class for week number 13. And let me tell you that it's a pleasure to have you here with me today. And I appreciate a lot the effort that you're doing into entering our online lessons and completing all of the assignments successfully that we have given to you in the previous weeks. So one more time, thank you for the great effort you're doing and keep it up. I'm pretty sure that with time we're going to adapt a little bit better to this way of working. And of course, you're going to be developing even better. Okay, so let's start with some general instructions. Get your physical book, enter to the Google Classroom, download your Word document, and remember that your Word document must have this format in its name. The only difference in this week is that you need to add week number 13 at the beginning, okay? And as we move, remember to take notes on the Word document. That is going to help you a lot to keep track of the most important information from there. And of course, it's going to help you out to learn even better. And this week, we're going to be learning about lesson number 1, pages 108 to 113. And our end in mind for the week is describes the structure of DNA and how it functions in genes. So this is the first thing that you need to keep track in your Word document. So if you need to, you can pause the video and come back later when you already have it in there. Okay, so this week we're going to talk about the genetic code. And we have already talked about DNA, about genes, about Mendel. But today we're going to go a little bit deeper into this topic. So, there was a DNA debut in 1951, and it was given by a very important scientist for genetics. Her name was Rosalind Franklin. And she discovered DNA can exist in a wet and in a dry environment. And she took the first picture of DNA by exposing it to X-rays. So this is a picture of Rosalind Franklin, and her discoveries were impressive to all of the scientists in 1951 because once he started exposing DNA material to X-rays, she noticed that the structure of DNA was like had a structure that reminded scientists of a letter X. So later on, they started studying a little bit more about it, and they discovered that it was a double helix. But that was one of the first discoveries of what the actual structure of DNA would look like in reality. And everything's thanks to her. Okay, so now we have how it forms. So now we have two other very important scientists, which were James Watson and Francis Crick in 1953. They published the first 3D model of what the structure of DNA looks like. So in the last part, I mentioned that the first image that scientists got was a picture that reminded of a letter X. But with all of the investigation that James Watson and Francis Crick did during the year of 1953, they were able to represent a 3D model. And they found that proteins can determine traits. Remember that traits are like certain characteristics from your body, like the color of your hair, the color of your eyes, if you're tall or short. All of that is determined by the proteins that you can find in the DNA. And DNA makes chromosomes. And DNA has a double helix structure. And something very important that you have to remember is that right in the center of DNA, or in between the double helix structure, you're going to find nitrogen bases. And basically, you are going to find 4. You're going to find adenine, thymine, cytosine, and guanine. So I already gave you a small introduction in class about these nitrogen bases. But basically, they are going to pair up. And they cannot pair up like randomly. Remember that adenine is only going to pair up with thymine, and cytosine is only going to pair up with guanine. So, and here's a picture of what a 3D model of DNA looks like. So, James Watson and Francis Crick developed the first 3D model. And I remember that they used different metal pieces, and they used different balls with different colors, so they could actually represent everything from the structure of DNA. Okay, and now we're going to analyze the patterns. So notice that there is a pattern. First of all, we have the DNA, which will be like the smallest structure in between the three of here. Then, after DNA recombines and gets together, then it is going to create a gene. And after a lot of genes are created, then they are going to create a chromosome. So that is the pattern that is always going to be followed whenever DNA is getting together to form bigger structures. And this is something pretty interesting. So several hundred millions of nitrogen bases can be found in a single gene. So imagine it's a gigantic amount of information that the body has to process in order to create one gene. And even it has to process even much more information to create a chromosome. Okay, and here I have a small pattern of what it will actually look like, according to the patterns of the DNA that can be found in the structure. So remember that we have four nitrogen bases. We have adenine, guanine, thymine, and cytosine. So here, all of these letters might seem a little bit confusing, but in reality they are not. They are just patterns of proteins or nitrogen bases. So letter A will state for adenine, then we have G for guanine, we have T and T for thymine, CC for a double cytosine, and then we go back to adenine and adenine. So basically, there is a huge amount of information in DNA. And the way you're going to find it in there is going to be in this structure or in this pattern. You are going to have the nitrogen bases in a different form or in a different order. And depending on the order that they contain, they're going to generate a different trait. Okay? And this can be found in all body cells, in exception of blood cells. So let's say that you are going to analyze a cell from your skin. It is possible to find nitrogen bases. It's possible to find DNA inside the nucleus of the cell that we are analyzing. Let's say that we're going to analyze one of the cells from your heart. Then it is possible to find DNA information in there as well. Okay, and here we have a small picture that represents what we were talking about before. So first of all, we have the nucleus in the cell where all of the DNA information is found. So pretty much what happens is that at first we have DNA. When it recombines, then we're going to have a gene. And when several genes are combined, we're going to find a chromosome. Okay, so let's continue. Now we're going to have the order of the bases. Here you go. This is what I was telling you about. So here you can see the combination of the nitrogen bases. And as you can see, all of the adenines that you can find in there, like the red ones, are only matched with the T's or with the green particles from there, which state for thymine. They cannot recombine in any other way, because if they do, then it's possible that a mutation will be generated in the body. And also, as you can see, we have letter C in yellow and letter G in sky blue, and they only recombine within each other, which are pretty much cytosine and guanine. So one more time, they cannot recombine with any other nitrogen bases, because if not, a mutation can be generated. Okay, so that's pretty much the nitrogen bases that we find inside the double helix. But what is this double helix composed of? So as you can notice, there we have two parts, which are the orange part and the purple part. So the orange part are pretty much deoxyribose, which is pretty much sugar. And also we have phosphate. So in phosphate and deoxyribose, when they recombine, they are going to be generating this double helix where the nitrogen bases can recombine. Okay? And now we're going to jump to our evaluation. It was a pretty quick lesson, but we have a very small content to analyze. So now read the pages from the topic 108 to 113 and complement your summary. So especially read pages 112 and 113. So I already summarized most of the content from the topic, but still I need you to go and read those pages so you can have a refresh on your mind to understand the topic a little bit better. So once you are done with your summary, you have to create a PowerPoint presentation with the information from there. Very important, please do not copy and paste the information. So I already shared my PowerPoint presentation with you, but that doesn't mean that you have to copy and paste directly from here. What you have to do is read and use your own words to show me what you have learned and understood from the topic. Notice, if you copy and paste, you are not going to have any points from the presentation. So use your own words better. So let's continue. You have to include your name, grade, section, topic, and end in mind as an introduction to your presentation. That is very important, and that is the first thing that I'm going to be checking in your PowerPoint presentation. So after you have finished with this information, you can go ahead and add the most important aspects from the topic. But at first, I need your personal information. Remember that you have to add images and transitions as well as animations. And what is the rubric that I will use to evaluate your understanding from here? So, completing meaningful content, 2 points. Images, 2 points. Grammar, 2 points. General structure of the presentation, that you have an introduction, a body, and a conclusion, 2 points. General appearance of the presentation, 1 point. And lastly, animations and transitions, 1 point. So that's pretty much the lesson for this week. Thank you guys for coming, and I wish you the best of luck when creating your presentation. If you have any questions, you can let me know in the Google Classroom or in the live meeting that we're going to have in Zoom. So that'll be pretty much it, and goodbye guys. Take care.